Okay, here we go. Chemistry 102, exam 4 review. Remember that exam 4, okay, so exam 4 is going to cover chapters, it's going to cover chapters 16, which is the carbohydrates chapter, chapter 17, which deals with lipids, and it's also going to cover chapter 18, which deals with protein. So let's get started with chapter 17. We've already looked at some chapter 16 problems earlier on, so we'll start with the first question, which is, which of the following is not classified as a type of eicosanoid? And if you looked at my video lectures, I spoke about eicosanoids in detail. Can anybody tell me what the prefix eikos stands for before we actually answer the question? Can anybody tell me what eikos means? Exactly. Like Johnny says, it's 20 carbons, right? The eicosanoids, which are molecules like thromboxanes and prostaglandins and leukotrienes, they all have 20 carbons in their skeleton. But there's one of these that does not have 20 carbons in its skeleton, and that would be steroids. Steroids are, in fact, not classified as eicosanoids. And as far as the prostaglandins, thromboxane, and leukotrienes go, we don't really cover the nomenclature in detail of any of those. What I asked you to be able to do in my lecture videos is to be able to recognize if you see a prostaglandin or a thromboxane or a leukotriene. Okay, just be able to kind of recognize the carbon skeleton. And of course, all organic chemistry students have to be able to recognize the skeleton of a steroid, which has an A, B, C, and D ring system like that. All steroids have that skeleton. Okay, let's move on to question 2. What response is caused by the prostaglandins produced in the kidney? Well, if you look at the function of prostaglandins, they have many different biological functions. If you see the first one, stimulation of ovulation, that probably doesn't have anything to do with the kidneys. Smooth muscle contractions, neither. Whereas dilation of renal blood vessels, these are blood vessels that are found in the kidneys, are in fact the response of those dilation of those renal blood vessels is caused by prostaglandins in the kidneys. Question number 3, which of the following statements concerning lipid functions is not true? So there's one of these statements that is maybe not true. The first one, let's read through all of them together as a group. We have A, which is the human body uses triglycerides for long-term energy storage, insulation, and protection. Well, that's absolutely true. Plants and animals use waxes as water barriers. If you think about, the expression water off a duck's back, the reason why water runs off of a bird's feathers is because of a waxy coating on feathers. The same thing applies to a tree leaf. If you've ever been in a rainstorm, and looked at water on a leaf, it usually forms these bubbles, and that's because there's a wax coating on the leaves. The next one, phosphoglycerides are key components of cell structure, of the structure of cell membranes. Definitely, this is true. Phosphoglycerides are all over the place in cell membranes. Steroids function as messenger molecules that enable cell communication. We know that that is true as well. And so in fact, all of these statements are 100% true. Let's look at a question that deals with structure. Question number 4 says, cetyl palmitate is a component of spermaceti wax isolated from the heads of sperm whales and is used in cosmetics. In which solvent would you expect cetyl palmitate to have the greatest solubility? So here is the molecule in question, cetyl palmitate. Now, what's the functional group that you see in cetyl palmitate? Could anybody tell me what functional group we see there in cetyl palmitate? Yeah, absolutely, right? There's an ester right here. Okay, so this is an ester. And whenever we have any kind of ester that has 2 R groups that are long chains like this, then we have a wax, right? That's what a wax is. Now, even though there's no carboxylic acid in there, only an ester, even though we have an ester, and you know that there's a dipole moment between the carbon and the oxygen here, and we know that this is a polar bond as well, Sure, those are polar bonds, but look at this. The hydrophobic portion of a wax, like these long alkyl groups here, are really big, and those are very, very hydrophobic. Both of those groups are hydrophobic. And so is this molecule going to solubilize in something like acetic acid, which is a little tiny acetic acid, which is very polar? Absolutely not. The same reason it won't solubilize in ethyl alcohol. Right? These are small polar molecules, acetic acid and ethanol, whereas petroleum ether, which it tells us it's a mixture of liquid alkanes. Alkanes are nonpolar. That takes us all the way back to chapter 10, the first chapter we studied in Chemistry 102. Right? Alkanes are made from carbon and hydrogen only. Okay? And so if we have petroleum ether, that's going to solubilize a wax like acetyl palmitate because it's mostly these big hydrophobic groups. It's absolutely not going to dissolve in water or solubilize in water, and it's not going to solubilize in a 5% sodium chloride solution either. Okay? In fact, waxes are going to definitely not be soluble in water. Well, let's look at the next one, which deals with human plasma lipoproteins. We looked at 4 classes of lipoproteins: the chylomicrons, the VLDL, the very low density lipoproteins, LDL, the low density lipoproteins, and HDL, the high density lipoproteins. But it's asking us which class of human plasma lipoproteins carries TGCs or triglycerides from the intestine to other tissues. And so you're expected to have a bit of an idea about the function of each one of these lipoproteins, and it's in fact the chylomicrons is where the lipids start, right? They take the lipids that are, packaged through the intestine, and then it carries them through to other tissues. So the answer to that one is chylomicrons. All right, let's talk about another question that involves structures. Testosterone, cholesterol, and progesterone are all classified as steroids because they contain a steroid nucleus. Which of the following compounds would be classified as a steroid? Again, you don't have to be able to draw a steroid from memory, but can anybody pick out, which of these molecules are steroids, if any of them? Absolutely. So Greg and Johnny, you both have it, right? It's A, or the first one, right? Because as I told you, all steroids have this A, B, C, D nucleus. We always find that. Okay, you're always going to find a 6-membered ring with another 6-membered ring attached to it, like that. That's a 10-membered double ring, but anyhow, we have another 6-membered ring, and then we have a 5-membered ring like that. And we call this the A, B, C, D ring system. Even though this one is aromatic, it still has the 6, 6, 6, 5 ring system in that order. And so the answer is A, which is the first one only. This one doesn't have the system, and that one doesn't have the system either. So those are not steroids. In fact, I have an uncle who was a pharmacist for 30 years or something, and I remember him coming to my dorm one time to bring me something when I was in university, and I was studying organic chemistry. He said, "Oh, wow, organic chemistry, I had to take that." He said, really the only thing I remember is what's the structure of a steroid, because when I see them as a pharmacist, I have to be able to recognize those. Okay, the next one, a tube of lipstick lists Capric Triglyceride among its ingredients. Capric Triglyceride is a simple triglyceride containing capric acid in a 10-carbon saturated fatty acid. What's the structure of Capric Triglyceride? Let me try to walk you through this one a little bit here as a group. Okay, what is a triglyceride, right? A triglyceride. So a triglyceride is basically a fat, right? And what is it made from? It's made from glycerol. So we take glycerol, which is this molecule, 1, 2, 3 propane triol. So this is glycerol, and then we combine it with fatty acids. Okay, we combine it with a fatty acid. Anyhow, the tail is not important because this is just my example. And we have to combine it with 3 of those because we're going to form ester bonds here, here, and here. All right, so that when you combine those, you end up with this. You have your glycerol backbone, right? You have a carbon here, right? This is a CH2. You have a CH here, and you have a CH2 here. Oxygen, oxygen, oxygen. Then you have one of the fatty acids here to make an ester, okay? You have another one here to make a second ester, and you make a third one here. And if you're wondering, hey, is a triglyceride just 3 esters attached to glycerol? Absolutely. But this is a triglyceride. Okay, now it tells us that it's made from it's a triglyceride, so it's got to possess the glycerol, right? It's got to possess that. You can see we have a portion of glycerol here, we have it here, we have it here. This one does not have it, so that can't be correct. And this one doesn't have it, so that can't be correct, right? You've got to have that glycerol backbone, so to speak. Then you're going to have a carbonyl, right? You've got to have a carbonyl attached. So we have a carbonyl. Actually, we have all these carbonyls here, so that looks good. We have all these carbonyls here, and we have all these carbonyls here. So all of these, A, B, and C, they all kind of have the structure of a triglyceride. But what does it tell us? It tells us it comes from capric acid, which is a 10-carbon saturated fatty acid. And so if we draw the structure of capric acid, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This would be capric acid. If you don't believe me, look at the carbons. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Well, now that you've had a chance to think about it while I'm rambling on here, could anybody tell me which one of these would be correct? Would it be A, B, or C? Brianna, you're 100% correct, right? Why would it be B? Well, there's got to be 10 carbons. Look, and I'm going to delete the red circle here. Look at B. You can see that this portion here has 1 carbon here. It's got a total of 8 carbons here, and then it's got this methyl on the end. So 1 plus 8 plus 1 gives you 10 carbons. Hopefully you notice there's a major problem here, right? You have another oxygen here. So that's not even a functional group that we've looked at. It's actually called a carbonate. So that is definitely incorrect. So we can cross that one off. Okay. And the problem with A, there's a big problem with A, is that it has a total of 11 carbons. Look, you've got 1 here, you've got 9 here, and then you've got 1 here. So 1 plus 9 plus 1 gives you 11. Okay, and it's not 11 carbons. And so the answer is, in fact, B. I know I did a lot of scratching around on this one. Give me a thumbs up if you follow me on the rationale here for the triglyceride being B. All right, wonderful, great, awesome. Let's keep moving. Good practice. Okay, another question that deals with a wax. It says when palmitic acid, a 16-carbon saturated fatty acid, reacts with a 30-carbon unbranched alcohol, a wax is formed. What's the structure of this wax? Remember, I told you earlier on this evening that a wax is nothing more than an ester. Okay? A wax is nothing more than an ester, except these R groups are really big. Right? This one is going to be really big and long, and this one's going to be really big, and that renders it insoluble. Okay, look, it comes from the wax comes from a 16-carbon saturated fatty acid. Look, we could draw it. Look, it would look like this. Okay, it would look like this. 1, 2, 3. So this is where my carboxyl. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, like that. All right. So there's a 16-carbon saturated fatty acid, and then we have a 30-carbon unbranched alcohol. Remember, that this portion of the ester, the part that's near the oxygen with only single bonds, that part comes from an alcohol, right? This part comes from an alcohol, and this part here, this comes from a carboxylic acid, Okay, so the alcohol portion is going to have 30 carbons. Look, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30. Woo, just made it. So there's our wax. Could anybody tell me which one of these looks like the most reasonable answer? Let's see, I'm getting some answers here. Okay. Yeah, the answer is D. Absolutely. Right? The answer is D. And the reason why is because from our carboxylic acid portion, okay, it's got to have a total of 16 carbons. Look, you've got 1, 14, and 1. You take 1 plus 14 plus 1, that equals 16 carbons. From the alcohol side, you've got 29, and then you've got a methyl group. So that equals 30. And so the correct answer is D. The problem with C is what? Is that the carboxylic acid portion here has 1 carbon plus 16 plus 1. So 1 plus 16 plus 1 gives you 18. So it's got too many carbons in it. Right? There are 30 carbons here in the alcohol portion, but that is why the correct answer is D. Okay, let's move on from there. Talk about sphingomyelin. Sphingomyelins are a type of sphingolipid, cell membrane lipids that contain sphingosine, an 18-carbon amino alcohol, as a backbone of the structure. What type of bond joins the fatty acid portion of the sphingomyelin molecule to the sphingosine backbone? Well, remember that a fatty acid, okay, a regular old carboxylic acid is something like, acetic acid. Okay, acetic acid is just a regular carboxylic acid. When do you get a fatty acid? When you have a long hydrocarbon tail. Okay, if we have an 18 carbon, or sorry, a fatty acid rather, it's going to have a long hydrocarbon tail. Could anybody tell me what the answer to this question is? Where do you see a portion that would have come from a fatty acid in this molecule? There's only one spot in the whole molecule where there's a portion that would have come from a fatty acid. In what type of bond? Yeah, Johnny, absolutely. It's an amide bond, right? Sorry, none of the space here. Yeah, the answer is amide bond. Good. Both of my students answered it correctly, right? This part here is the only part that could have come from a carboxylic acid because it's the only part with a carbonyl. But you know that when you take a carboxylic acid, right, and you combine it with an amine, you can form an amide, right? And so that's what you see is an amide there. Okay, so carboxylic acid plus amine gives you amide, and that's what you see right here is this is an amide bond. All right, let's move on to number 16. It says tristerin has a melting point of 72 degrees Celsius. Would it be classified as a fat or an oil? Could anybody help me out with that one? It doesn't melt until 72 degrees Celsius. That's pretty hot. Doesn't melt until you heat it up to 72 degrees Celsius. Would it be a fat or an oil? Could anybody answer this question? What would it be at room temperature? Would it be a solid or a liquid? Yeah, I see that I have 2 students who answered fat. Absolutely, because at room temperature, so we'll put at room temperature, fats are solid, oils are liquid. Okay? If tristerin doesn't melt until 72 degrees Celsius, that means that at 25 degrees Celsius, tristerin is a solid, therefore a fat. Boom. Okay, let's try number 11. It asks us to draw the products that are formed when the following triglyceride is hydrolyzed with aqueous sodium hydroxide. So we're going to take this triglyceride and we're going to heat it up with aqueous sodium hydroxide. Right? Could anybody tell me the name of this reaction? It's got a sassy name that we learned. Take an ester and you treat it with sodium hydroxide. Absolutely. It's a saponification, right? Hydrolysis of an ester in base. Right? How do we know we're hydrolyzing an ester? Because we got 1, 2, 3 esters right here. And so we're going to break all of the carbon-oxygen bonds when we treat this with sodium hydroxide. What's that going to produce? Well, you can see that from this portion of the molecule, I have 1, 2, 3 carbons, and they all have an oxygen. Looks like glycerol to me, right? So I'm going to have this guy right here. 1, 2, 3. So I'm going to form glycerol. And I'm also going to hydrolyze all of those esters back to carboxylates, right? Remember, sodium hydroxide is a base. So we're not going to form carboxylic acids. We're going to form carboxylate salts, and that's how soap is made. Let's count the number of carbons on the first two. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So the first two are identical. The last one is not identical because it's got a cis double bond in it. So that means we're going to get 2 molecules of this carboxylate. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And they're going to be sodium salt. So we're going to get 2 molecules of that sodium salt, of that fatty acid. And then this one here, oh, good gravy. Do I have to draw that? Let me try my best here. I'll probably miss a bone or something. 1, 2, 3, 4, 5, 6, 7, 8. Okay, close enough. There we go. So we'll end up with this sodium salt as well. And there you go. That's what you're going to get. You're going to end up breaking all those carbon-oxygen bonds, and you're going to end up with carboxylates. Remember, you wouldn't end up with carboxylic acids because you're in base. Okay? And again, this is what soap is. This is how the pioneers made soap. And that's all soap is. It's nothing more than this. Okay? Hydrolyze animal fat and base, and you, my friend, end up with a soap. That's why we call this saponification. You can write that in there as well. This is a saponification reaction. All right, let's move on. Number 12. This one I would never ask you. This is more of a discussion point. I'm never going to ask you to draw a lecithin, not in this lifetime anyway. It says a lecithin was hydrolyzed to yield 2 equivalents of myristic acid. Draw the structure of the lecithin. Again, I would never ask you this one. But I'll draw it for you because a lecithin looks like this. It is covered in my lecture slides, but I would never ask you to just draw this, out of thin air. That's not really fair. So we have 1, 2, 3, and then here we'd have this. We'd have like that. There we go. And then in the lecithin, you're going to have this phosphate ester here. So I'll put this right here. All right. We have this piece from choline. Anyhow, my question to you is, and I think this is a reasonable question for my students. This compound is chiral. Identify the chiral carbon. Could anybody describe to me what a chiral carbon is? Let's say I highlighted. Well, no, let me just ask you if you could describe that to me over Teams. There is one chiral carbon in this molecule. No? Okay. Remember that for a chiral carbon, okay, it has to be attached to four different groups. So not four different atoms, it's got to be attached to four different groups. All right? And so the chiral carbon is in fact this carbon right here. Okay, it's that one that I have highlighted in red. Why? Because it's attached to a hydrogen. Okay, so it's got the hydrogen. It's got this ester attached to it here. It's got this ester that has a CH2 attached to it. That's different than the other one in blue. And then it's got all of this group down here attached to it. And so the chiral carbon is the one that I have highlighted in red. All right, the next one is a conceptual problem, but I think it's a reasonable problem. These are kind of questions where I just like to get my students to think a little bit, you know. Octanol is more efficient than hexanol at crossing a cell membrane and entering the cell. Explain. Well, if we draw the structures of octanol and hexanol, 1, 2, 3, 4, 5, 6, 7, 8. There's octanol, and hexanol will be 1, 2, 3, 4, 5, 6. This is, hexanol. Could anybody explain to me, or just, you know, give me a short unmute of their mic and give me some kind of reason why it might be better? Octanol might be more efficient at crossing the cell membrane than hexanol. Go ahead.
Yeah, is it because the octanol has a longer hydrophobic chain, allowing it to pass through the hydrophobic region of the cell membrane?
Absolutely. Couldn't have said it better myself. Okay. So in fact, let's try to write that down so everybody gets the answer there. So we'll put it like this. Octanol. Thank you, Johnny. Octanol has a longer hydrophobic tail. Than hexanol. And is therefore more efficient at crossing the nonpolar environment of the cell membrane, right? Because the cell membrane, and I'll just draw it up here. I'm sure if you've taken a biology class, you've all seen this before, where you have all the hydrophobic or hydrophilic heads, and then you have all the hydrophobic tails coming off of those, like this. There should be two on each one, but Mr. Dion isn't the world's greatest artist, right? And then you have all the hydrophilic heads over here in your phospholipid, right? Okay, well, all those hydrocarbon tails that point down there, those are all nonpolar. Okay, this is all nonpolar. And so in order for a molecule to efficiently cross through the cell membrane, it has to be nonpolar enough to get across. Let's see. I don't know, this is kind of a fun one, number 14. Would you expect glycerol to be able to cross the cell membrane? Anybody have an idea about this one? It's not meant to be a trick question. It's more just kind of looking at the structure of glycerol and thinking out loud about it. Do you think glycerol could cross the cell membrane? Based off of what we just talked about? Just a yes or a no is good enough for this guy. Yeah, Johnny says no. I agree, and so does Stephanie. I agree with you. The answer is no. Glycerol has 3 hydroxyl groups, which are extremely polar, and no hydrophobic tail. Okay, and therefore it's not going to be able to cross the nonpolar environment of the cell membrane. This question is only for the people who tried this ahead of time. I had this posted in an announcement. Did anybody try this problem and try to classify these PGs, these prostaglandins, according to the substitution patterns found in your slides? I would never ask you to come up with this out of thin air, not in 100 years. This is more just kind of an interest thing, if anybody gave it a shot. So if you look at my slides, and if you look at the lecture videos, I put up a slide. It doesn't even come from our textbook. I ripped it out of another textbook, but it explained where prostaglandin nomenclature comes from. So PG stands for prostaglandin. E stands for the substitution pattern on the 5-membered rings here. And then the subscript, the number, stands for the number of double bonds. Okay, so both of these would have to have something PG something, 1, and this would have to be PG something 1. Anyhow, in case nobody went ahead and took a look at it, you can go back and look at my slide, but this first one would be a PGE1, and then this is a PGF1. Again, that was nothing more than, something just for my students who wanted to look at the structures and see if they could figure them out. I'll never ask you to pick those out of a police lineup or whatever. This next one here is also a question that I took from another textbook. Well, we can take a quick look at it. It says here, the following compound was isolated from nerve cells. Describe how this compound differs in structure from fats and oils. Well, the first thing I noticed is that this compound does have a glycerol backbone. So in case you're wondering, it does. Here's the glycerol backbone. I'll circle it in blue. You can even see they put a wedge here because that's the chiral carbon. So glycerol, over here, glycerol backbone. Well, what's the difference? Not backbone. The difference is that a triglyceride, which comes from a triglyceride that's found in fats and oils, has 3 esters. Look, we have an ester here. We have an ester here, but over here, this is not an ester. So that's how the structure of this compound differs from fats and oils. 3 products are obtained when you hydrolyze it. Anyhow, don't worry about drawing those. 4 products are obtained when you hydrolyze it with aqueous acid. Again, that's kind of beyond the scope of our class, so I'm not going to ask you anything about that. You can just cross those ones out. I took those from, believe it or not, from a second semester full year organic chemistry class. So anyhow, all right, well, let's switch gears and talk about amino acids and peptides and proteins for a little bit. Some chapter 18 problems. I don't know, I think I put some tricky ones in here too, or not tricky, but some interesting problems. I might have to mess around with a few of these to make them a little bit easier, but anyhow, some fun problems. Okay, well, let's get started. The thing is that your instructor, sometimes I try to come up with questions and I'll go to books that are, more complicated. But again, I'm going to tell you the bits that you need to know. All right, everybody good on chapter 17? Ready to start chapter 18? All right, great. I want to make sure my students are still there. Perfect. Okay, let's do it. Okay, first question. Amino acids are the building blocks of proteins. Which of the following statements concerning amino acids is false? There are 20 different amino acids that occur naturally in human proteins. This is true. The amino acids that make up human proteins are called essential amino acids. That is, in fact, untrue. That is false. right? Remember, there are essential amino acids and non-essential amino acids. The non-essential amino acids are ones that we can synthesize in our body, but essential amino acids are amino acids that we cannot synthesize. So we can put here amino acids we cannot synthesize. Okay, so that's what essential amino acids. What else? The amino acids present in human proteins are alpha amino acids. Yes. Except for glycine, where R is equal to H, amino acids are chiral. Yes. Amino acids are classified by the nature of their R group. Absolutely true. Okay, so the answer is B. Move on to number 2. What's the term that we use to describe the amino acid that has a free carboxylate group in the peptide? I went over this with you last lecture that we did online. Can anybody tell me? We have our NCC and our NCC. What do you call the end that has the free carboxylate group hanging off of it? Yeah, Stephanie's 100% correct. We call it the C-terminus, right? Or the C-terminal, which is the way our textbook describes it, right? So if you have, an amino acid, a tripeptide, let's say you have NCC, 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 right? By convention, on the right-hand side, we put the carboxylate, and we call this the C-terminal, okay? And on this end, we put the protonated nitrogen, and we call this the N-terminal, like that. Okay, and you can fill in the rest of the molecule if you want, I hope you've all been practicing drawing your alpha amino acids so they have different R groups like this. Anyhow, you get the idea, so we call that the C-terminal amino acid. All right, what are the two most common types of secondary structure found in proteins? I'm sure that somebody can nail this one pretty quickly. Absolutely. Thanks, Stephanie. Yes, the answer is C, the alpha helix and the beta pleated sheet. Remember that primary structure is actually the order. So it's the order of the amino acids and the amide bonds that hold them together. So amide bonds. Secondary structure, right? That's alpha helix, beta pleated sheet. Beta pleated sheet. Right, and then tertiary and quaternary structure were the result of hydrogen bonding, hydrophobic interactions, salt bridges, and disulfide bonds, right? And that was when we had the folding in of our amino acid, right, is the tertiary structure. And when we have two or more proteins with the tertiary structure, rather, that's when we have quaternary structure. Okay, number 4. The amino acid 5-hydroxylysine is found in collagen, which represents the structure of this unusual amino acid. Well, in order to complete this problem, you'd have to have the structure of lysine with you. And so if you needed the structure of lysine, I would provide it. Excuse me. But the structure of lysine looks like this. So we have our carboxyl end, right? We have our this end here, and then we have 1, 2, 3, 4 CH2 groups like that, and then we have an NH3 plus. Okay, so the question becomes, which one of these is the 5th carbon? Well, we always start numbering at the most oxidized carbon, so this would be carbon 1, 2, 3, 4, 5. Since this is the 5th carbon, that is where our hydroxyl group will be. It will be on this carbon, which, if you look here, we have 1, 2, 3, 4, 5. So this one here, number 3, would be 5-hydroxylysine. Again, I'll just remind you that if you erase this and put a CH2 here, this would be lysine, which is a basic amino acid. Okay, number 5, proteins are long chains of amino acids joined together by peptide bonds. Which of the following properly depicts the peptide bond formed between two just generic amino acids? I'll wait for my students to answer this one. We looked at this enough. Remember, when we put two amino acids together, we go NCC, NCC. We put our carboxylate on this end. We put our protonated amine on this end. It goes NC-carbonyl like that. We have hydrogens here. We have one on this nitrogen. And then we have our R groups like this. So that is the structure that we're looking for, something like that. If you look at A, it goes N-C-carbonyl, N-carbonyl, right? That's not correct. If you look at B, it goes N-C-carbonyl, N-C-carbonyl with a carboxylate. Everything else looks correct. And so this is actually the correct answer. Let's take a look and see what the problems are with C and D, because there are some issues there. So in C, it goes NC nitrogen. Remember, it has to go NCC. Son of a gun. Let me do it. NCC, NCC, like that. So that's incorrect. And if you look at D, it goes NC carbonyl. And no, it's got an oxygen. So that's not correct either. Look at E. It goes NC carbonyl. No. Got to go NCC, NCC, like that in that order. And so the correct answer is B. Okay, number 6, tufsin is a tetrapeptide that stimulates and promotes the destruction of tumor cells. What is meant by a tetrapeptide? A tetrapeptide. What would a tetrapeptide? Let's see. Sorry, I'm going to scroll down here. So if we go back to our last problem and we look at what I drew here, when we have two amino acids linked together, this is a dipeptide, right? Because I have two amino acids linked together. And so what Brianna's saying is C is that a tetrapeptide is when you have four amino acids joined together by peptide bonds. And that's what a tetrapeptide is. Right, if you look at A, it says there are 4 separate peptide chains. No, a tetrapeptide is just a peptide put together with just 4 different amino acids. If you look at B, 4 peptide bonds join the amino acids together in a structure. No, it can't be that either, because in a tetrapeptide, look, I have N-C-carbonyl, N-C-carbonyl. I have N-C-carbonyl and N-C-carbonyl, like this, okay? How many peptide bonds are there in a tetrapeptide? 1, 2, 3. There's only 3 peptide bonds in a tetrapeptide, right? There would be 3. The 4 different secondary structures are present. No, it's got nothing to do with it. This protein has quaternary level. No, it's got nothing to do with it. So the answer is C. Okay, number 7. What is meant by quaternary structure of a protein? I'll let you guys take a look at it. And there's one answer that's definitely better. It's head and shoulders above the rest. Yes, you're both correct. Stephanie and Brianna, both correct. The answer here is E. I wanted you to take a look at those. So it says, what is meant by quaternary structure? It describes the association of two or more polypeptide chains to form one functioning protein unit. Remember, for a protein that has quaternary structure, it has to have the quaternary structure in order for it to be functional, right? Otherwise, it doesn't, won't be able to form its biological function. Okay, number 8, consider the following forms of the amino acid alanine. Which statement concerning these structures is false? I'll give you a second to take a look at the answers instead of me reading them all. See if you can come up with the right answer. All right, I got a few different answers here. Okay, it says, so remember, we're looking for a false statement. So D, it says structure 4 represents the zwitterion. This is correct, right? This is correct. This is a zwitterion. What else do I have here? I got E. None of the above statements are false. Well, let me help you out with this one, okay? If you watched my videos, hopefully you would have seen that, in fact, the one that is true is false, is A. It says structure 1 represents the form of alanine present in blood at physiological pH. The structure that represents alanine at physiological pH is going to be the zwitterion, okay? This is what you would have at pH equal to what, 7.4 or whatever. That's physiological pH. It's going to exist as the zwitterion. Okay, so the answer is A. Let's move on to question 9. What is the term for the state of a protein whose organized structure has become completely disorganized? I give you 5 seconds to solve this one. You completely disorganize a protein. What's it called? It is. Yeah, it's denatured, right? You've denatured it. Okay, good. The next one, I also give you 5 seconds to answer it. I'm not a speed guy, but number 10, what protein is the oxygen storage protein in muscles? Yeah, and in muscles, it's myoglobin. Muscles, myoglobin, right? Blood, in the blood, it's hemoglobin. Let's take a look at the next one. I mentioned this briefly the other night with you guys. I talked about it kind of quickly, but we looked at a structural protein just for a second. Can anybody pick out which one of these was the structural protein that I mentioned the other night? It can't be albumin, right? That's egg protein. Yeah, it's keratin. It's keratin. Keratin is a structural protein. You might remember that I looked at the microfibril, then the macrofibril, and how they were put together to make keratin, and then it showed a hair fiber. Hey, question 12 is a good question. One of my favorites. Which term best describes the relationship between L-alanine and D-alanine? Yeah, thanks, Stephanie. Absolutely. It's enantiomers, right? Enantiomers, non-superimposable, mirror images, like that. Perfect. Thanks. That's awesome. The next one shows a oh, I'm giving away the answer. Yeah, it's asking us which of the following statements are false. I'll give you a second. Again, to answer this one here, you'd have to have a cheat sheet that showed you all of the natural amino acids, which I will provide if you need it. But could anybody answer 13? Which one is false? Okay, let's take a look. So the first thing, it says it is a tripeptide. That's true, right? Because it goes NCC, that's 1, NCC, that's 2, and NCC, that's 3. So it is a tripeptide. The first one is a glycine, the second one is a proline, and the third one is serine. So this is also correct. There are 2 peptide bonds. Yes, there's a peptide bond here and a peptide bond here. Remember, if you have a dipeptide, it's only got 1 peptide bond. If you have a tripeptide, it's got 2 peptide bonds. If you've got a tetrapeptide, it's got 3 peptide bonds. If you have a pentapeptide, it's got 4 peptide bonds. So it's always got 1 less peptide bond. D, the carboxyl group of the proline is joined to the amino group of the serine. Here's the carboxyl group of the proline, and it is joined to the amino group of the serine. That's correct. The last one says E, the peptide contains only hydrophobic amino acid residues. On serine, you have a hydroxyl. This is hydrophilic or water-loving. Okay, so that is not true. Okay, that is in fact false. So the answer to 13 is E. Let's see here. Number 14, remember what type of attractive force is responsible for maintaining secondary structure, right? That was the alpha helix and the beta pleated sheet. What holds those together? Yeah, thanks, Brianna. You could just answer that with 2 words, right? Hydrogen bonds between the carbonyls of the amides and the amines, and that's what holds it together. Nothing more than that. Secondary structure. Okay, number 15. Some of these questions I might need to help you out with. Which of the following best describes where the amino acids isoleucine and tryptophan are most likely to be found in the tertiary structure of a protein? So let me help you out with this, okay? Let me ask you this question a different way. The 2 R groups in isoleucine and tryptophan, would you say those are hydrophobic or hydrophilic? Right, they're both hydrophobic. Absolutely, both are hydrophobic. Okay, so then did anybody watch my videos and is able to answer this? Yes, Johnny, you're 100% correct. Right, so what happens is that when you have a protein and it folds in to give you tertiary structure, all of the hydrophobic groups are going to be found on the inside. So the hydrophobic interior of the protein. And the reason why is because these groups are hydrophobic. What's the exterior of the protein? What's the environment going to be? It's going to be aqueous, right? The aqueous environment of the cell. And so any polar R groups are going to be found on the outside, but anything that's hydrophobic like these, those are going to fold in on the inside to get away from the aqueous environment that the protein is in. And so the answer is E. Okay, let's try number 16. And I might need to help you out with this. Let's just keep it simple. Let's do alanine. I'll change this up a little bit. Let's do alanine at a pH of, I don't know, 12. And let's do
Mr. Dion,
yeah,
you circled E, but I think you meant to circle D.
Did I? Yes, you're right. Sorry, Johnny. Thanks. I was testing you guys. In the interior, folded away. from the water due to their hydrophobic. Thanks, Johnny. Thanks for pointing that out. Let's try, if we try to draw alanine at a pH of 12, and then let's try to draw, I don't know, let's draw tyrosine. So let's forget about B for now. Let's draw tyrosine at a pH of 2. Okay, so let me help you out with these. If you had, you know, a cheat sheet that had all the amino acids, okay, and it drew, it had alanine on it. Alanine's going to look like this. It's going to give you this bitter ion. And alanine, the R group on alanine is simply a methyl group. So this is at pH 7.4, physiological pH. I'll draw the structure of tyrosine. So again, this is alanine. Tyrosine looks like this. Okay, if you haven't looked them up already, tyrosine looks like this. So we have CH2, we have an aromatic ring, and then we have a hydroxyl. So here's what all I really expect you to know. Okay, if you were to take alanine and put it in a pH of 12, is a pH of 12 acidic or basic? Not a trick question. Is a pH of 12, would that be strongly acidic or strongly basic? It's going to be very basic, right? The higher the pH, what did I just do? Basic and acidic. I'm not sure what I just did there, you guys. Anyhow, is it everywhere? Son of a gun. Let me try. Whatever. You can still see it. So what I'm doing is I'm taking that amino acid and I'm putting it in a strong base. Am I going to decarboxylate here? No, it's already been done, right? So there's no acidic proton to remove there. But is there an extra proton I can remove here from the ammonium? The answer is yes. This is a weak acid. And so when I put that in a pH of 12, pH equals to 12, what it's going to look like is that my carboxylate is still going to be the same, but my ammonium will have lost a proton. Son of a gun. And so it's going to look like this. So overall, the charge is going to be -1. If I take tyrosine and I put it at a pH of 2, that's strongly acidic. All right, so that means I'm going to protonate the carboxylate, and I can't add another proton here. And so when I put that at a pH equal to 2, what I'm going to do is I'm going to protonate the carboxylate, and I'm still going to have my proton on my amine. There you have it. Okay, again, the next one, you need a cheat sheet to do the next one. Again, if you need that, of course, I'm going to provide that to you. I'm not going to leave you out in the cold, you know, struggling to figure out the names of the amino acids. Let me just kind of walk you through it, how you would do it. We have NCC. Here's our first one. Then we have NCC. There's our second one. Then we have NCC, like that. Then we have NCC. Then we have NCC. And so this is a pentapeptide, right? There's 5 amino acids in here. Where are the R groups? This is an R group here. And this R group comes from leucine. So the first one is leucine. Leucine. Okay. The abbreviation for leucine is LU, L-U. I'll never ask you about one-letter abbreviations. The next one just has a methyl group on it. Hopefully you picked that one up tonight because we've seen it a few times when we have just a methyl group. That is going to be an alanine. So we have ALA. The next one, we have a phenyl group attached to a methyl. So this is phenylalanine. And for that, we use PHE, phenylalanine. The next one is cysteine. We have a CH2 and a thiol. Cysteine, like that. So we use CYS. And the last one comes from aspartic acid. Aspartic acid, or aspartate, is the conjugate base. So there we go, ASP, like that. Again, I would never ask you to pick these out unless I gave you a cheat sheet where you had the structures of the amino acids. You don't have to memorize the amino acids in Chemistry 102. But you should be able to pick out the fact that this is a pentapeptide. Right? You should be able to identify where the R groups are. Also, you don't have to memorize the 3-letter abbreviations for all the amino acids. If I gave you the structures and you needed the 3-letter abbreviations, I would provide those for you. This next one here, we'll ignore that. Let's try this one. Okay, let's try number 20. Yeah, we could look at number 18 quickly. Number 18 is kind of a weird question. I took it out of another textbook. It says, determine which of the following peptides will have a higher molecular weight. So a higher molecular weight. Hint, it's not necessary to actually calculate the molecular weight of each peptide. You just compare the side chains. So for cysteine, we have CH2, SH, right? That's the same on both of them. SH. For tyrosine, we have CH2 with an aromatic ring and a hydroxyl. For leucine, we have CH2, CH2 methyls, like that. For phenylalanine, we have CH2, and then we have a phenyl. And then for isoleucine, we have CH3, CH2, CH3. So you can see that these two parts are identical. This part and this part aren't identical, but they both have 4 carbons and 6, 7, 8, 9 hydrogens. So those are going to have the same weight. So how are you going to differentiate between the two? The only difference between this one and this one is this one here has a hydroxyl. So since that one has a hydroxyl on it, it's going to weigh more. And so this one will be heavier by 1 oxygen and 1 hydrogen. So that's around 17 grams per mole. Right? Oxygen weighs around 16, hydrogen is 1. Okay. The next one, I'm not going to ask you about that. So let's try question number 20. It says, methionine enkephalin is a pentapeptide that's produced in the body to control pain. From the sequence of amino acid residues, draw a bond line structure of methionine enkephalin. I'm going to give you a second to see if you can draw this. So I'll give you a few minutes to take a look at it. All right, let's take a look at how we would solve this. Remember, it's a pentapeptide, so that means we've got 5 amino acid residues, or 5 amino acids all linked together. And so we're going to start with NCC, 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 and NCC. You might have had to Google each one of these 3-letter abbreviations to get the R groups, but let's figure out what the rest of the molecule would look like. We're going to have our carboxylate on our C terminus. We're going to have our ammonium on our N terminus. Remember, it goes NC carbonyl, NC carbonyl, NC carbonyl, C carbonyl, like that. Each one of these carbons in between is going to have a hydrogen attached to it. And the nitrogens are all going to have hydrogens attached to them. So what's missing? The only thing that's missing now is an R group here, 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 and here. 1, 2, 3, 4, 5 R groups. So the R group for our tyrosine is a CH2 with an aromatic ring, followed by hydroxyl in the para position. For glycine, it's the simplest amino acid. The R group is simply a hydrogen. We have 2 of those in a row. Next, we have phenylalanine, which has a CH2 and an aromatic ring attached directly to it, like that. And the last one is methionine, which you probably had to look up. It goes CH2, CH2, sulfur, and CH3. So it's a sulfide. Fix my sulfur. And there you have it.